Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing another really awesome guest today, creating a better tomorrow for all of us. On the show today, we have the honor of being joined by Dr. Michael McCullar, who is the CEO of RegCell. They are a biotech company. They have developed this really interesting, best-in-class proprietary platform that allows for the generation of epigenetically reprogrammed antigen-specific regulatory T cells, or Tregs, that are ultimately stable in vivo, as well as have a high degree of functionality to restore immune tolerance and ultimately reverse autoimmune disorders, as well as have a lot of applications in organ transplantation. Dr. McCullar has a long history of experience in both biotech and pharma development, over 2 decades worth. Most recently served as the CEO of OnQuality Pharmaceuticals. They are providing leadership to that organization that was involved in targeted oncology supportive therapies and developing pioneering medications really to address the intricacies of various cancer treatment-related toxicities. And before that, he was the chief operating officer at Tolero Pharmaceuticals. He had a really important role there, and ultimately the development and then ultimately the company's acquisition by Sumitomo Dainippon. Several other roles at organizations like Astex Pharmaceuticals, Supergen. Dr. McCull holds a PhD in toxicology from the University of California, Riverside, went on to an MBA from the W. Carey School of Business at Arizona State University, and a lot of really cool topics to get into today, especially due to the fact that their founder just received the Nobel Prize a couple of days ago. So very timely, very relevant, and we're honored to have him. Dr. Michael McCullar, thank you so much for taking time to come on the show today.
No, it's my pleasure. Thank you for having me.
Yeah, awesome having you, and a very exciting time, needless to say, for you and the company. But I'm really looking forward to getting into all these themes with you. Before we touch on anything related to Tregs and autoimmune disease and so forth, would love to start off with you, Mike, and just hand you the floor for a few minutes to talk a little bit more about your background. I mentioned you had a PhD in toxicology and did a really fascinating set of work in terms of biodegradation of all sorts of nasty chemicals. Went on to do chemo drug development work in MSRA and antibiotics. Tell us a little about you.
Thank you for the question. My background's a little bit different, though. I really am a toxicologist. At the time, I was interested in chemistry and biology and also in math, and I couldn't tolerate sight of blood. So I couldn't, for me, it was a way to think about pulling those pieces together. And it was a great experience. You learned a lot. But when I got out, I worked for about a year as a toxicologist. I didn't really like it, and was fortunate enough to be recruited to join a company and work in regulatory affairs for a few years, which I think as a young professional, it's a great experience, really, to be exposed to how the FDA works. And after a few years, the company needed help in manufacturing. And I felt that, well, I'd done tissue culture, right? So I could do that. And I didn't know anything about it, but it was a great experience because in the process, you learn many things, and also how important it is and how hard it is truly to make advanced therapies. Those are early days of antibody manufacturing and also early days of cell therapy production and gene therapy vectors. So it was a great experience for me, but I really didn't see myself as having a career in manufacturing. So I was recruited to join another company, where I really was fortunate to be given a responsibility to lead an NDA for, at the time, was a very novel drug. So then again, as a young professional, being exposed to how to launch a phase 3 study, how an NDA gets put together, the complexities of how these parts work and how the FDA processes, it was a wonderful experience for me. But at the time and after that, the company I felt was really transitioning. And we had an approved drug, a small commercial sales force, and the company sent me to business school. And there was a great experience because I think, when you earn a PhD, you very much get rewarded, acknowledged what you do as an individual. But in business school, learn more about how to collaborate. And it became clear, the company was struggling a bit with identity and having a competitive advantage. And then it became clear to me, we were kind of a medium-sized company, and I really felt we wanted to develop a unique set of skills that give us an advantage. And there you could see at the time, small companies were really doing a great job at competing with larger companies, how they use technology innovation.
Sure.
So we really wanted to, and that really led to this acquisition of Astex Therapeutics at the time, which gave us really what I felt was a world-class, or what we think of as market or category leading type of skill set in research. And when you put that together with that we had an approved drug and clinical infrastructure, we became a very interesting company. So it didn't take long after that. I had become SVP of business development, and we were to get acquired by Otsuka for those capabilities. And it's interesting because that was my first exposure to Japan, really.
Mm.
And I got to look at Japanese science, Japanese culture, Japanese management systems, and it was a wonderful experience. But after a year, I went on and we licensed a drug with some friends of mine from Sanofi, and that became Tolero, and we tried to do the same thing. These ideas of, to us, what's important around differentiation and advantages in category leadership. So we were able to license a drug from Sanofi and start to or think better about it in terms of how to build that into a really novel, differentiated story. So it didn't take long. We were acquired by Sumitomo Dainippon Pharma.
Hmm.
And then I was a little bit more senior, and part of the deal was we all agreed to stay for 4 years, and it was a great experience and to still see more about Japanese science, the quality of Japanese science. And it was great experience. But after 4 years was that people were scattering everywhere, and I had taken another job. About a year into that, maybe about 3 years ago, I was approached by a Japanese venture fund called UTech, and they had this really great, interesting idea. And the concept was, Japan, as we know, it creates world-class science. You can just see from last week, there were two Nobel laureates emerging from Japan.
Yep.
So it doesn't really matter how we measure it. Japan, in terms of Nobel laureates or Nature papers or IP, it really ranks usually second to the US, but is yet to develop really what we think of as an ecosystem to enable these emerging companies to reach their full potential. And the idea was, why don't we take the best in class Japanese companies, but we don't want to separate them from Japanese science. There's a major advantage for doing scientific research in Japan, but enable them to access these resources that may be scarce in Japan that limit the ecosystem. And if we could do that, we could really unlock a lot of value for patients by globalizing what we think is best-in-class science. So we always felt that RegCell represented, best-in-class Japanese life science opportunity. And that was the view. So last year and a half or so, we've been working pretty aggressively in that vision. And this to us was a wonderful surprise. We thought really reflects the quality of science. And the emerging from Japan is obviously Shimon Sakaguchi is really, really unbelievable, not just scientist, but just an unbelievable, inspirational person. He's really amazing to work with. So it's been a great experience.
No, it must be, no doubt. And again, before we get to, some of the discoveries that he made, let's lay, the landscape
Yeah.
here because, we routinely talk about unmet medical needs on the show, and
Mm.
here are the two sort of major ones. We have autoimmune disease on one hand, hundreds of millions of people worldwide, hundreds of billions of dollars. And obviously the audience is familiar with your rheumatoid arthritis and
Mhm.
lupus and
Yeah.
psoriasis and stuff like that, less
Yeah.
so scleroderma and
Mm.
Sjogren's and
Yeah.
Graves and everything else, but a major packet of unmet medical needs globally. And then transplantation.
Yeah.
Again, a multi tens of hundreds of billions and just in terms
Yeah.
of rejection drugs and all the
Mhm.
processes going on there.
Right.
And a sort of unified sort of therapeutic basket. I mean, some things that overlap in terms of your steroids and your, general immunosuppressive drugs,
Yeah.
and then drugs that, are a little more specific in terms of your inflammatory cytokines and other
Mhm.
targets. Let's talk about this giant basket because it's
Yeah, that's
huge.
great.
It's maybe a unified unmet medical need we don't think about, but it's there
Yeah.
and it's big.
Yeah. Yeah, that's for sure. So We always believe that any good company starts with a clear problem to solve. So first thing that we wanted to do with RegCell was just take a step back and ask this kind of a macro question about, what problems are we solving. Then it became a little easier to put the context around this question. And what we look at is we've been treating autoimmune diseases for about 75 years, right? And they're with blunt instruments that broadly suppress the immune system. They don't offer curative potential, which means that patients usually take them chronically, sometimes require lifelong therapies with these drugs. And oftentimes, even what we think of as the most contemporaneous agents still have box warnings because of serious side effects,
Yep.
elevated rates of malignancies, serious cardiac toxicities, elevated mortality. And really, none of them get to the root cause of lack of self-tolerance. And none of them can really distinguish a good immune cell from a bad immune cell. That's what it boils down to. And so we felt, and Shimon Sensei always believed, his view was, can we use Tregs to restore antigen-specific self-tolerance and with durable outcomes? So we felt at the core of this, and the reason why Tregs become so interesting is that they are indispensable non-redundant cell type to maintain self-tolerance. So when they become impaired, that's a hallmark of autoimmune disorders. So our view was, could you leverage the Treg concept to deliver antigen-specific self-tolerance? And at the time, it's impossible to do that with existing therapies. And the interesting thing that people sometimes maybe don't appreciate is that autoimmune disorders are not caused by usually one autoreactive antigen, usually multiple, and many times they're unknown. So targeting unknown antigens becomes a very complex question. So his idea was actually extremely innovative. He thought, well, maybe the best way to target unknown or complex antigens with a Treg is to start with these autoreactive CD4 cells, because if you can convert them to a Treg, the T cell profile could be maintained. Then the Tregs could actually copy the autoantigen fingerprint to be activated by the same antigens the bad T cells are. But that was the concept, but no one knew how to do it. So, ultimately, we learned quite a bit about this process, how to do that. And that's really the foundation, I think, of, our company, driven largely by Professor Sakaguchi around this concept and how to do that.
And it's interesting because Dr. Sakaguchi's
Mhm.
work, when he initially
Yeah.
finds these properties of regulatory T cells back in 1995, and it's
Mhm.
always interesting to see how the work from getting the Nobel Prize happens.
Yeah.
But
Yeah.
that's another topic. But the really interesting thing
Mhm.
about them in terms of, okay, we have this interesting population of cells normally that maintains tolerance to self
Mhm.
antigens, tries to prevent autoimmune disease
Mhm.
normally. The way it happens is, again, as you were just saying, kind of combinatorial and very
Oh, yeah.
complex in
Yeah.
terms of and I'm going to simplify this. These
Yeah.
Tregs go over there and say, hey, here's some anti-inflammatory cytokines, and here's some,
Yeah.
I'm going to block these growth factors, and I'm going to interrupt these antigen presenting things. They do a lot of stuff. And,
Oh,
and
they
I
do.
wonder if
Yeah.
you could
They
just
do.
talk about sort of like what the Tregs are normally doing in our
Yeah.
bodies,
Yeah.
and
Yeah.
then we'll get into what you're doing with them.
Yeah. So Tregs, as I mentioned, we think of them and they've been described as a non-redundant, indispensable cell type. And they have very complex pharmacology to modulate autoreactive T cells and autoreactive biology, including not just cytolytic type effects, but maintaining metabolic control, right?
Mm-hmm.
Antigen presentation, they modulate that. So they're very sophisticated. And Shimon Sensei has this famous little figure he sketched out about the complexities and how they do that. So they can suppress not just autoreactive T cells, but they also suppress autoreactive B cells. People may not
Okay.
appreciate that. So that's why we think that they are really a key regulator of self-tolerance, because they can do such complex biology, not just mediating autoreactive T cells, but also the maturation, development of autoreactive B cells. So we have those data coming out in a very nice paper in a few weeks, we hope, that will articulate that and also bring together about the platform science, but also the translational aspects of it that are very compelling. So that's part of it, is we want to solve two problems with our platform. One is what we think of as misperception that Tregs are inherently unstable, but also the ability to achieve antigen-specific type of suppression. So they're there, they are essential in terms of maintaining self-tolerance with this biology. And one way to think about it, to make it a little more simple to strategize around, is almost like enzyme replacement therapy, right?
Sure.
in the sense that you have this fully functional cell type that effectively maintains cell tolerance. And when they become impaired, that effectively is really a hallmark of autoimmune disorders and somewhat of in terms of how the disease develops and progresses. So in that context, if you restore Treg function with, say, a technology like ours, then patients should get better. That's how we think of it and how we think about looking for correlations with impaired Tregs and pathology so we can think more about where to leverage the science. Right. So that's how we think of it. And it's an easier way because it's very complicated, right? So somewhere we need to kind of simplify this concept. So we think of it in many ways like enzyme replacement therapy.
Mhm. And then there's one other
Yeah.
side point, and then we'll get into the sort of the technology.
Mhm.
The the ability to generate immune tolerance occurs both centrally in the
Right.
thymus, in the bone marrow, but also
Right.
peripherally. And I wonder if
That's
you could
right.
just say a few words, because I think people think, wow, I got rheumatoid
Yeah.
arthritis in my
Yeah.
knee. I
Yeah.
also got a thymus gland. I got bone marrow. Just say a couple words about the dynamics of how
Yeah, well,
things travel around in this.
central tolerance responds in the thymus, and it's like a checking, and it's an interesting way to think of it. So we all have in the background, all of us will have the immune cells that mistakenly recognize our own antigens as foreign, and will have the potential to develop an autoimmune reaction. Usually, the ability to check self-tolerance is checked in the thymus before immune cells release. And that's pretty effective. But when they do, then we get this population in the periphery floating around that can detect, or they actually mistakenly identify our own antigens as foreign. And usually that's where Tregs become important because they will control these autoreactive bad T cells. But when they get broken, right? Tregs become impaired, then these autoreactive T cells in the periphery, they will expand and enrich, and then they become disease causing. So the main goal for Tregs is in the periphery. They're around in tissues and sometimes migrating in blood, interacting with other cells to check and make sure we can control these
Mhm.
autoreactive T cells in the periphery. And that's, I think, why we think about Tregs the most, is in the periphery, not necessarily in central tolerance, but in peripheral tolerance.
Got it. Got it.
That's a simple way. I hope it worked.
No, no, perfect. No, spot on again and again, because I want to again take a little time to walk the audience through each
Yeah.
of these because it is really interesting process, and that gets us to a little bit of what
Mhm.
an antigen specific Treg now is, or
Yeah.
one that is
Yeah.
epigenetically reprogrammed. Because
Yeah.
again, in my mind, and on the show, we profiled CAR T cells, we profiled
Mhm.
epigenetic reprogramming.
Oh,
It
good.
seems like you're
Yeah.
using a little combination of both here, a little of the magic in
Yeah.
the sense
Yeah.
once, okay,
Yeah.
we're going to CAR T CAR T sort of our cells. So hey, we're going to go after that a type 1 diabetes
Yeah.
antigen,
Oh,
that
yeah.
multiple sclerosis antigen, and at the
Mhm.
same time, you're doing some epigenetic magic that says, hey, in this fascinating milieu of the immune
Mhm.
system, little things can, little stimuli, let's say, can change events, and you're making the cells more stable than sort
Yeah.
of they
Yeah.
traditionally are. So again, I mean,
Yeah.
again,
Yeah.
take your time on this
Oh,
one
sure.
because
No, no,
I
this is
really want
great. This
to
is great.
understand it. Yeah.
Yeah, it's a wonderful story, and I think it's just fascinating. It's a little bit fortuitous too, right? Serendipitous in many ways. So, as we go back to when Shimon Sensei published his paper around the discovery of these rare T cells that had this different function to maintain self-tolerance. It didn't take long till after those papers came out when people started to imagine the importance of Tregs, and you started to see papers come out from investigators where they may take a patient with autoimmune disorder, maybe like type 1 diabetes, and try to isolate that patient's Tregs, try to expand them and reintroduce them. That's kind of the basis of autologous cell therapy.
Mm-hmm.
And those experiments didn't really work out very well. And I think it makes more sense that we know more about Tregs. And if you use this kind of enzyme replacement concept, that
Mm-hmm.
taking Tregs from somebody who has type 1 diabetes, for example, and trying to expand those, it's likely there may be some impaired function in those. And Tregs don't expand very well. When you expand them, they really, there's some features that make them prone to apoptosis, for example, or
Got it.
unable to recognize bad antigens. So it doesn't really work that well, which is, but they are safe, right? So we learned quite a bit from those experiences. And in the meantime, immunologists learned about this idea, what we describe as induced Tregs. So if you take a naive T cell
Mm-hmm.
and treated with TGF-beta IL-2, they'll adopt these suppressive features. People call these induced Tregs, but they don't adopt really a Treg epigenome, so they're inherently unstable. So they don't really make good living drugs. And that's when we wondered, or Shimon Sensei wondered, well, maybe the best way to have antigen-specific self-tolerance with a Treg is to start with these bad T cells, because then you could retain the TCR profile. So the resulting Tregs could copy the same antigen fingerprint, stimulating these bad T cells. Then we could perhaps achieve antigen-specific suppression. But no one knew how to do that, right? So, especially with an activated T cell. So we just did some basic screening of small molecule libraries, but with activated T cells. And in this experience, we came across a small molecule compound that could turn up high levels of FOXP3.
Okay.
We didn't know how it worked and didn't know as much about it, but it turns out this compound really interferes with what's called the mediator complex, and that's important in terms of epigenetic control, and also is known to be involved with chromatin remodeling.
Yep.
And as you know, chromatin remodeling is a very powerful epigenetic process that dictates cell fate.
Yep.
And it's important in Treg biology. So this compound was wonderful in terms of a step forward about taking an activated T cell and adopting some Treg biology. But it turns out it wasn't enough to get what we really needed to really reprogram fully these what we think of as Treg imprinting features that lock them into a Treg phenotype. So the next question we asked was, what happens inside the thymus, right, when Tregs are developing from precursor cells into Tregs? What happens to lock them into a Treg phenotype, right?
Mm-hmm.
It turns out inside the FOXP3 gene, there are these non-coding regions that are super enhancers. And you might imagine also that in, say, a conventional T cell, these suppressive type functions need to be silenced. And obviously, epigenetics plays a big role in silencing genes. So it turns out within the FOXP3 gene, these epigenetically silenced regions, where you learn just from emulating in the thymus, how the thymus they get reprogrammed to lock them into a Treg phenotype. So we learned how to emulate that. So then we were able to, one, restructure the chromatin in a way to make this region accessible, then emulate what happens in the thymus to demethylate specifically these non-coding regions that are involved in Treg imprinting and locking them into a Treg phenotype. And when we could do that, then we could even reprogram functional genes up, you might imagine also, in a conventional T cell that doesn't want to turn on Treg functional genes. They're also epigenetically silenced. So then we can learn how to demethylate the promoter regions to emulate a Treg. So now we've reprogrammed through multiple essential epigenetic processes and pathways to then lock them into a stable Treg phenotype at the chromatin level, at the super enhancer level inside the FOXP3 gene, and then demethylate the silenced promoters that control expression of functional genes. So in this, we've been able to take a bad T cell and then reprogram it into a very highly functional, lineage stable Treg that now maintains the same TCR profile. So they respond to the exact same autoantigens driving autoimmunity, even unknown antigens. Are cases where autoimmune is driven by multiple antigens, which is highly differentiated and novel compared to other types of approaches.
Sure. And obviously, in using, okay, you're starting and you're doing this amazing reprogramming process and targeting process. Clearly, this leads into the importance of autologous manufacturing and personalized, Treg therapy. Can you just say a couple of words? I mean, obviously, we talk a lot about cell therapy from different perspectives on the show, from stem cell transplants to parabiosis and everything in between. Just for the audience sake, what is involved in the context of
great
how
question.
how many engineered Tregs would I require with my Graves' disease versus my wife with her psoriasis, or what? You talk a little about that.
Yeah, that's a great question. So a couple of things to keep in mind that are important for us. I didn't talk about it earlier, but the first thing I wanted to think about when I became involved with the company is, what do we need to do to ensure we can create, as the backbone, a commercially viable process? And some things we have in our advantage that are important. One of those, we don't require gene editing, so the complexity is not, we can eliminate the complexity there. And that really simplifies the supply chain for sure. And the other part is we don't require viral DNA or anything for transduction, say, for example, with a car. And the good part about that is one, it reduces complexity, but really makes the process more amenable to automation. And our view is automation is a major driving factor to scalability and containing costs. So that's a huge advantage, we think, for us, and it's played out really very well in terms of how we interface with the FDA and how we think about scalability and engaging with US-based CDMOs and attracting the right kind of talent. That's a huge advantage for us in our view. And so what we think also about what's important for our cells, and I didn't get a chance to talk about it earlier, it's quite novel, and you'll see this in a paper coming out. So the reprogram we do is very specific to the FOXP3 related biology and these things that lock them into a Treg phenotype. So they don't necessarily change all the genes, which means there's some features of our Tregs that maybe I think are retained from the effector cell function. One of those has to do with the chemokine receptor patterns on the cell surface, just like a bad T cell. It's very different. And that is important because that enables them to be extremely good at homing to inflammatory sites, effectively chasing the bad T cells to their inflammatory sites where they're causing disease. And what's also you'll see in the paper, which really is important, is that our reprogrammed Tregs from effector cells, they do tend to be better in terms of suppressive features and also at homing to inflammatory sites now where they're challenged with these autoreactive antigens, which drives their expansion and in vivo persistence. So when we look at our data, it's not a drug, right? Like you would think of a small molecule drug, it's not a static number. And so they tend to be very good at homing where they enrich and expand. So we don't see a huge dose response because they do increase in numbers at the inflammatory sites. So when we think about dose response and safety, it's a little different for us. So we think, for example, several hundred million cells, we hope to have a good effect, but we may learn more about that when we launch this first human study where we evaluate several doses.
So yeah, I mean, speaking about that, I mean, and again, there's limited information that I could pull, but clearly, as I mentioned at the beginning, organ-specific autoimmune diseases, huge basket, organ transplant, graft versus host, and I guess host versus graft also huge area. And then obviously, sort of this third segment, allergies, and which is, the whole principle there, immune tolerance as well. Can you talk a little bit about some of the initial clinical indications that are being discussed? I went through some of Dr. Sakaguchi's papers, and obviously he's studying a lot of things, but take us down this path of what you
no, it's a great question. And we spend a lot of time with this concept. And when I first joined the company, we learning how to explain this, and we would talk to people, and we able to articulate the value proposition and how novel this is, and the transformative potential of the concept in the biology. So people would be very excited about it, and then they would say, "Well, that's excellent. So how will you do this in the clinic?" So I spent lots of time, doing quantitative work during my career, and this concept about indication selection, when I hear that in my mind, I'm thinking, well, you're really asking me to solve 10 questions at once.
Right.
And it all starts really fundamentally with getting the science right. And then we have to answer other questions in our minds about, value is not created always by the epidemiology of a disease, by the problem you're solving. So we're wondering these questions: where do broken Tregs drive pathology? We're looking for huge gaps of efficacy where there isn't effective treatments. And we also need to think about enrollment. We need to solve this question for market access. But the one important point that really is important in our view, I think sometimes people overlook this, is we are needing to add value fast. So part of our concept about delivering value in indications is we need unambiguous endpoints, right? That we can measure easily, that are not ambiguous. Right. So we're trying to look for indications early on around these ideas, but with endpoints that are easy to interpret. So we're trying to avoid long-term endpoints that are subjective or composite, or involving complex biopsies or involving complex types of imaging. And so that's how we thought of it. And that's really the basis of how we were able to secure this, we think is a substantial non-dilutive grant to get us to clinical proof of concept, is to solve all these things and deliver value in a way that makes sense to investors, but to patients. So we can do a lot of interesting things and take big swings, but first of all, we have to solve these fundamental concepts, almost like a stepping stone, right? Can you prove certain things early and then enable us to add value and solve problems, but then take a bigger swing at something like type 1 diabetes or MS or RA. So that's how we solve this. And I think you'll hear more about that. We have a lot of patents that are being prosecuted around methods of use and those things, but pretty soon we'll be able to talk about that in more detail.
Mm-hmm. Mm-hmm.
But once we do that, then we can think about things about transplantation and things about other larger questions. But first of all, we have to solve these fundamental questions about the platform and how to get that into patients and how to solve some really, we think, are pressing, overlooked, and substantial unmet medical needs evolving where we think broken Tregs are playing a problem.
Yeah. Yeah, the other interesting thing, and it's going to be interesting again. We'll probably come back and do another show at some point. But I noticed that Dr. Sakaguchi had quite a few papers, interesting on sort of because regulatory T cells play a really interesting part in cancer.
Oh, yeah, it's a whole
this whole cancer immunotherapy issue.
That's right. Yeah, that's
CAR
we
Ts
have
and all that.
Oh, yeah, yeah. It's a great question, and we do have an effort and how would we describe that? Progressing effort and one way to describe the company, we don't talk about it a lot because I think it's confusing to some people. And, we're very focused on getting this first in human study done, but there's a whole nother side of Treg biology that we're exploring around their implications in cancer immunotherapy resistance.
Yep.
Yeah, so we think we have something very important there. It's just been, we're not that big of a company, so we need to focus on this. So there's a whole nother side that we're working towards in that space. We just haven't been able to focus on it as much.
No, I understand that. I was just saying again, it's he has a rather fascinating array of papers out there, and when you start thinking about, wow, there's a lot.
No, it is, yeah. No, it's a
there's a
huge,
lot to that.
yeah. It's really, I think we're on the very. And one interesting thing about working with him is when we generate so much data, and usually it's very much on the cutting edge of immunology. So our challenge is, how do we keep up with these ideas? One thing we need to make sure we're doing in the company is fully comprehending this emerging science coming from our labs, and how do you leverage that in the most impactful way?
Yeah, I mean, I joke around, but I for one find the immune system more complicated than the brain. Honestly, it's with all the things that we're learning, about all these different types of cells and
I know.
functions.
I feel the same way. I'm not an immunologist by training, right? So I have to learn so much, and it's just being part of it. I think when lucky, because I tell this story all the time when people ask me, but when I got a PhD, I was in a lab with a famous guy, and I had a fellowship, and he really could care less if I was there or not, right? So I was so excited. I show up, move there early, and I'm maybe 2 weeks early or a week early, and I go into the lab to say hello. And he just told me, "Oh yeah, I forgot you're coming, right? Well, here's your desk." He just looked in the corner. That was it. So part of, I think, what was good for me was I had to learn how to learn autonomously, right? And I wasn't a biologist. I was a chemist. These guys in the lab were all molecular biologists and microbiologists, and there was no one there to help me. And I figured, this is not great, and I just want out. The only way to get out is to do. But I had to learn myself how to learn autonomously. I think that was maybe the most important point I learned from my education was autonomous learning. And so here, the same thing. I'm not an immunologist, but having to learn what this means, how to interpret data has been a great experience. So it's wonderful. And he's such a patient and engaging and committed person. It's been really wonderful.
Awesome. So so I mean, to sum up so far for the audience, antigen targeting, in vivo stability, tissue homing, better safety than a lot of the nasty stuff we currently use for immunosuppression, and then the manufacturing edge. Could you, we'll talk about sort of the U.S. Japanese
Yeah,
transition,
yeah.
but could you say anything you could say about because I know there was a press release about a partnership with a group called GeneFab, which is working with you on the
Oh,
scale up
yeah.
and the GMP. Where is RegCell right now in terms of
That's a great question. Yeah, great
FDA
question.
meetings,
Yeah.
pre-IND
I'm
stuff,
glad you
whatever?
asked. Yeah, we
Take us
just
down here.
we just had our pre-IND meeting with the comments back a few weeks ago, so that was great. And we normal. I think nothing. And we're very happy with the outcomes. A lot of I think what I felt for the pre-IND for us. I didn't feel going into it there was much controversy around the clinical design. It's pretty basic, right? There's
Mhm.
nothing special there. So they didn't have any. There were no red flags and just affirmations regarding how to identify patients, who's in, who's out, how long to track safety, blah, blah, and then pretty straightforward. Fortunately, we have a very comprehensive data set. So part of it was ensuring that there was clarity regarding our non-clinical GLP work, dosing schedules, and
Mhm.
how to think about any additional data we may want. So nothing special. I think that went very, very well. A lot of development work on the CMC. So that was very helpful to enable us to understand if there are any gaps or if what we're saying is going to be acceptable. So that went very well. So it gives us a very clear picture. We have a new CDMO in the US. It's going very well. So we think that enables us to be able to submit the IND we hope, towards the end of Q2 of next year. Right? So that's great for us
Mhm.
and gives us a very clear picture now. The execution is really on enabling us to launch those studies and working backwards. And that's, I think, going to be the biggest push and really where we've been working heavily the last year as we transition from we think, as a platform science company to a clinical stage company. And a few months ago, we were able to become a JLabs portfolio company, which has been tremendous because it gives us access to world-class expertise around functionality and skill sets we don't already have. So as we
Mhm.
think about, one question we're solving for is, market access and how to make sure we're doing that the right way. Making sure fundamentally we're building a commercially viable type of manufacturing process, which they're great at. And also, as we transition in our business plan, there are other indications we're interested in developing commercial plans for, and they're great for supporting those efforts too. So we have a wonderful relationship with them, and it's been very productive.
Nice. Um, yeah, so say also a couple words about the recent headquarter move. I mean, I know you wrapped up
Yeah.
a nice seed round plus the
Hmm.
non-dilutive stuff from
Hmm.
AMED in Japan,
Hmm.
and a lot of your investors were Japanese institutes or venture funds, part of the
Yeah.
universities.
Hmm.
Just say what's going to be happening in Japan still, what's going to be happening
Yeah.
in Emeryville.
Hmm.
Talk to us a little bit about that.
That's a fundamental question that we sorted out early on. And it
Hmm.
really gets to this concept I talked about early about, how do we ensure we can access the right types of capabilities that are scarce in Japan. So if you take a macro look at the ecosystem, some things will be kind of clear to us is, you know, pharma people, a lot of the experience in Japan in life sciences are still sequestered in Takeda Astellas.
Yeah.
And that means in Japan, in these ecosystems, trying to scale and develop and execute in companies like RegCell in Japan, it's these skill sets we need for clinical development, for example, are very, very scarce.
Mm.
So if you and I were to walk down in Boston and or over here
Right.
in San Francisco, every company would have a very sophisticated set of capabilities to think, contemplate, articulate, and execute a clinical development plan, right?
Hmm.
And in Japan, that's not available easily to small companies. And maybe one of the reasons why I think that the ecosystem has struggled, it's hard to do that. And they rely on different ways to solve those questions that maybe delays their advancement. But here it's a lot easier, right? So it's easier for us to do that. And one thing we did when we first moved to the US was develop the right cable network to enable us to, and the right talent to think about these clinical development plans, how to leverage the science.
Hmm.
So that should be, we think, in the US for now, clinical development for sure. And we were able to recruit world-class talent with commercial expertise in how to manufacture T cells. That's been hugely beneficial for us because I always felt that we need a commercially viable process and we need to be able to do this in a high quality, reproducible, effective way so we can launch and execute complex clinical trials.
Yeah.
That's much easier in terms of also capacity at CDMOs, and that talent is much easier to access in the US, right? So we think, this sophisticated CMC development, clinical development, that's easier to access in US, and that's what we're doing. But the fundamental platform development and discovery science, we have a major advantage in Japan. So that and we can't imagine doing that too far from our co-founder, right? So having the foundational science in terms of T-reg biology, how to leverage that, how to optimize it, how to solve science questions should be in Japan. And the other parts we think would be better to have in US because now we can access these scarce resources we think that enable us to reach the full potential is by doing these kind of cross-border efforts.
Mm-hmm. It's exciting.
That's wonderful. I always tell people, on the outside it looks easy. Of course, all companies, we have lots of things we want to do, and I always tell people, these are not easy jobs. But if we can have 5 great days and one bad day, we shouldn't complain, right? That means you're doing really well. So that's kind of how we think about it.
Right. No, really cool. Again, I'm gonna look forward to continue to follow what you're up to, and as this all transitions into the clinic. Like what else should we know about that's coming up? I mean, I know you're sort of out on the conference
Mhm.
circuit. I know a couple
A
weeks
little
ago
bit,
you
yeah.
gave a story of
Yeah.
RegCell. This was at Lifetime Venture Startup Elevate.
Yeah. Yeah. It was a great experience. Yeah. Yeah.
What
Yeah.
else is happening for as we
Well,
get to the
yeah,
end of
people,
2025?
yeah, people. I think, I couldn't talk enough about my experience with people. These companies are all about people, and
Right.
I do have a very strong view on how we access talent, how we develop culture. I mean, culture for me, when I first was learning about these things, that I was in business school, but I was working and, was practicing these ideas on culture and values on people, right? Doing deals, acquiring companies, and I was practicing these concepts in a very, very positive way. So we talk about culture values all the time. And it's easy to do in the US, it's complex in Japan. But the reason we learn it's important, culture values are kind of things that we use as words, but we're trying to really is get behaviors and outcomes.
Yeah.
But what is important is in my view, we're trying to get people engaged. Engagement's a huge thing for us and how we attract talent. And engagement is important now, I learned. It's not just what it's getting us is resistance or persistence and resilience. And that's what we're trying to accomplish with culture because these are hard jobs. And the biggest, I think, question to success is how can you get people to be resilient? So we're very aggressively starting to add key people around capabilities that we want. And, I don't think about companies as org charts. People ask me about org charts, but we really try to solve questions about capabilities. And now getting people in the company around the clinical development that can really bring world-class talent to make sure we're asking the right questions about how to optimize the platform in the clinic. And that's hugely important for us. We have a CMC team that's really great, but somebody, we're talking and thinking about recruiting a full-time CSO at this point. And what we're looking for here is somebody who really understands closer to what our data are saying. We're generating world-class science data. We need someone in the company who can understand and work closely with Shimon Sensei. So when they deliver information to us, the right people are inside the company to interpret the data and know how to use it.
Mm-hmm.
Yeah, so we're asking these very important questions and making sure we bring the right level of people into the company. The company's been a U.S. entity only for a few months, so we don't really have a huge team in the U.S.
Got it.
And we're very careful about titrating up the right kinds of talent because culture is important to us and execution is very important to us. And having the right people and the right fit who can understand what we're looking for in terms of advancing the science, but enable us to develop the right kind of culture so we can really be successful.
Got it. Awesome. So, Mike, one last question. I mean,
Mhm.
when Professor Sakaguchi won the Nobel Prize the other
Mhm.
day, it
Mhm.
seemed like my social, I guess I follow a lot of science accounts or whatever, but
Yeah.
my social media, like it blew up with everyone congratulating him. We want to
Mhm.
congratulate Professor Sakaguchi about these amazing
Yeah.
discoveries. So anything exciting happen in-house? I mean, do you have a
Yeah,
party?
well, I mean,
What
we're
went
always,
on?
yeah, we're a little giddy still. Everybody's still quite
Imagine.
excited, but we're going to celebrate. We'll head to Japan in a few, not too long. We'll have a nice event to acknowledge him. I can't, I was at that Lifetime Ventures event, and I can't. Maybe this is before he was acknowledged, but we're just really reiterating him as a person. He's not just a world-class scientist. He's an unbelievable individual in terms of his character and his generosity, his kindness. He just, yeah, it couldn't happen to a better person, well deserved. And we were just surprised, but not surprised, if you know what I mean. We always assumed I'll tell you an interesting story too, and I shouldn't say this out loud, but I'm not an immunologist. So when I first met him, I didn't fully understand who he was, right? And I would have really nice discussions with him in his office about the science and I was telling people what I was interested in doing. They said, "You know that guy?" I said, "Yeah, he's just a very nice guy." He goes, "Mike, he could win a Nobel Prize." I said, "Oh, awesome." But he's just a very humble, very kind-hearted, very generous, unselfless person. He's just really amazing. So, as I mentioned, we're extremely excited for him. And we'll do something together when we see him maybe in December. But he's quite active. He's on our board. I talk with him usually a couple times a week about the science and what we're doing, and questions and people and organizational items. And he's great, highly engaged with us. And I've always felt that he gives us really kind of a privileged set of capabilities that we can use to help our company in terms of how we create an advantage and fence off other types of platforms.
Absolutely. No, it's I'm excited for you.
No,
I have
yeah,
to
thank you.
say.
Yeah, that's why I said, these companies are ups and downs, and we always say if we can get 5 good days for every one bad day, we should not complain. So this has been a good week for us, right? We just brought in the first part of our pre-A financing and took down the second part of our grant. So it's been a good couple of weeks for us.
Good to hear. That's good to hear. No, it's again, I will I look forward to continue to follow the story. Hope to have you on again next
No, I'd
year
love to,
as
yeah.
things progress. But again, for everybody out there that's going to be listening to this episode of the show across the various podcast networks or who will be watching on the YouTube channel, you've been spending time with Dr. Michael McCullar, Chief Executive Officer, RegCell, doing really amazing work to develop this novel platform to ultimately help restore immune tolerance and deal with a lot of these unmet autoimmune disorders as well as limitations with transplantation. Mike, again, I want to thank you for taking the time out of the schedule to talk to us for a little while and walk us through what you're up to. Thank you for everything you do there. And as we like to say on our show, thanks for helping to create a better tomorrow for literally hundreds of
No,
millions of
yeah.
patients.
You're very
Really
kind.
awesome.
You're very kind. Thank you for the time. It's been a great hour. So thank you.
Yeah, great talking to you.